Hi everyone, welcome to Ethan's Sweet Corner. Today we are going to make my first ever creation. I call it the Organelle Graham Cake. This cake is enjoyable, surprising, and educational for both parents and kids because we are going to infuse all of these sweets into the cake so it can represent the different organelles in a plant cell. So I am just going to show you guys the stuff that we need for the cake. So first is the materials. Some plastic gloves, an 8 by 12 molding pan, an 8.5 diameter mixing bowl, and then a whisk, and then some tongs, and then a spoon. So now we need the materials for the cake base. So we are going to need 3 packs of condensed all-purpose cream, and which contains 250 ml each, and then 3 graham crackers which weighs 200 grams of, and then 1 can of Alaska condensed milk which contains 300 ml. So now for the cake toppings, we would need some gummy snakes and gummy worms which weighs 500 grams each, and then 690 grams of marshmallows, 168 grams of nips, and then 80 grams each of Zestar Sola, and then some popping candy, and then 130 grams of cream stick, and then one pack of pastillas. I also saved some sprinkles just in case you need them, and 1 pack of it is 25 grams. So now the first step is to put all of the cream into this bowl. By the way, you chill the cream 2 hours or less. So now we just cut it and then put it in the bowl. So now that we have put all of the cream inside the bowl, we are now just gonna mix it for 2 to 3 minutes. So now that we have mixed all of the cream, we are now gonna put in the condensed milk. So now that we have opened the condensed milk, we will now just put it inside our cream. So after putting all of the condensed milk, we will now just mix it for another 2 to 3 minutes. Now after we mix it, we now chill it for 2 hours. So now that the cream mixture is chilled, we will proceed in making the cake base. So the first step is to layer the pan with graham crackers. And then for the spaces, we will be dropping crumbled crumbled graham crackers. Next, we are going to apply a thin piece of cream or a thin layer of cream on top of the graham. So we will now repeat steps 1 and 2 until we run out of cream. We have used up all the cream for the top or last layer. We will now proceed in putting the cake toppings. The cream will serve as the cytoplasm. For the cell wall, we are going to use these marshmallows. For the cell membrane, we are going to use these green gummy snakes. For the vacuole, we are going to use Zola powder. Next is the Golgi apparatus, and we are going to use yellow colored snakes. Next is the nucleus. We need the popping candy for it. Nucleolus. We are going to use pastillas. Next, we are going to use purple snakes to make an endoplasmic reticulum. For the amyloplast, we are going to use a cut out purple snake head. For the mitochondria, we are going to use cut out pieces of red gummy snake. For the chloroplast, we have cut out snake gummies. For the peroxisome, we are going to use candy coated chocolate. For ribosome, we are going to use these candy sprinkles. So now we're done. This is my finished product, my Organelle Graham cake. It is best served when chilled. I hope you like it. Goodbye!